Thank you. Thank you very much. Thank you very much for inviting me, Cedric. It's really a pleasure to be here. And thank you all the graduate students who I just met half an hour ago. That was really interesting. I wish we could do those sessions on the camera live. It would be fun. Anyway, I'm going to talk today about what one means by differential expression. And I should say that actually, in some sense, this is the first part of a two-part talk. The second part, which I will deliver in genome informatics in about 3 weeks. So I think those are recorded as well. So if you're interested in what I talk about today, you might wish in a couple of weeks to go look at that talk when it comes online. The talk is actually work jointly with my current PhD student, Lin Yi at Caltech. She's a joint MD PhD between UCLA and Caltech. And my former student, Harold Pimentel, who's now at Stanford. And finally, my former student, Nicholas Bray, who's got a star because he's really a sort of co-corresponding author, if you like, on this project. He had a lot of the initial impetus for the project and many of the ideas. Also, I'd like to just acknowledge some other students and postdocs of mine and collaborator, Paul Melstead in Iceland. Vasilis is my postdoc, and Jay's a grad student. Because a lot of this project is actually not about doing things, but about thinking about things, and they're really helpful in that regard. So I'll start off with talking about actually a paper I wrote back in 2010, which is about RNA-seq. As you just heard, I've worked for a little while in this area, and this paper was the work of my former student Cole Trapnell, who wrote this program called cufflinks. For a while, you know, this even today, this is a program people have used a lot for RNA-seq assembly and quantification. But this paper, separately from publishing a tool, actually also published some ideas. And one of them that we really actually debated a lot internally was when you do RNA-seq, really should you be looking at genes or isoforms? And together with our collaborator, Barbara Waltz, who's now my colleague at Caltech, we came to the kind of philosophical, if you like, decision that we felt both the software but also the science should operate at the transcript level, because we felt that there's a lot, that's basically the unit at which biology happens, and that the technology was mature enough already in 2010 to extract meaningful information from that. And I've just highlighted here just a part out of the methods to show you that, you know, from the outset, we developed the method with transcript individual isoform quantification in mind. And we actually really tried to hammer home this point. We created a series of figures, which are like the one in the bottom right-hand here. We call them Napoleon plots because you might know this book by Edward Tufte where they show a lot of things on one plot, like the soldiers marching to Russia, and you can see the temperature and the weather and how many soldiers died and everything on one plot. And this bottom right-hand figure is kind of in that vein, trying to load a lot of information into one figure, and it's specifically isoform or transcript level information. So this is an example from our paper, which studied myogenesis. There was, Barbara had done an experiment, myogenesis RNA-seq with 4 time points, and MYC is an important gene in that process. It's got 3 isoforms. You can see that 2 share transcript start site, has a different site, but 2 that share a start site have a different exon. You can just see the yellow has a slightly different 5' end on its middle exon. And what we measured are these 2 pie charts at the right-hand side, which you can kind of ask about whether there are changes in transcriptional regulation by looking at the total quantification of A plus B versus C, or you can ask if there's post-transcriptional regulation going on, which is just a change in splicing with the same transcription start site, and that would be measuring A versus B. And in that plot on the bottom right-hand side, what's happening is we're showing for each pair of consecutive time points whether we have more of A plus B versus C than A versus B. And so blue is transcriptional and red is post-transcriptional. And we kind of wanted to indicate that during development, this gene is not only changing its overall expression level, which you see going down, but actually the dynamics inside are changing. And so anyway, I'm emphasizing this just to say that for a long time, I've advocated that RNA-seq is not just a replacement for microarrays that's maybe cheaper or faster or better, or, it's actually giving a window into new biology that was not really attainable, because even though microarrays, had splice array versions and so on, really getting this kind of information is very difficult. We made a lot of plots like this in the paper. Example I'm showing because this kind of thing come up later in the talk, where we actually saw complete isoform switching happening, where one is isoforms diminishing, the other is increasing. And if you were to look at the gene level, it's obvious in an example like this. If you just add up those quantifications, those abundances, everything's flat. So you really lose a lot of the picture of what's actually really happening underneath the hood of the gene. I think that we were somewhat vindicated in our philosophy over the years. There have been a number of papers showing that splicing is really important, in disease. It's useful diagnostically as a biomarker. It's a bit ridiculous to have a summary slide. There have been a ton of papers with really interesting stories about splicing, about isoform dynamics in various systems. And I think it's fair to say that transcript level at RNA-seq, with RNA-seq transcript level biology has been a thing and it's useful. But, a couple of, maybe 2 years ago now, a year and a half ago, my former student, Cole Chapnell, came to dinner at Berkeley, and we had a nice couple of drinks. And then he sort of whispered to me and he said, Lior, we've been bashing on about the transcript stuff all these years. And, we had these opposing team, people who did the other software like EdgeR, DSeq, they were saying, look, you shouldn't even do isoforms. It's not reliable. You should do genes only. And he said, I'm a biologist now. So Cole actually was a computer scientist. He was a student at the University of Maryland with Steven Salzberg, and he came to the math department at Berkeley and worked with me for 2 years. And then he transitioned to really do wet lab biology, and so he got to know the wet lab people, and he was doing it himself, and he said, my friends and colleagues, they don't care about transcripts. They care about genes. And the reason is that it's really hard to knock out a transcript. There's not a lot of technology for, isolating and knocking down one isoform of a gene. Now, maybe nowadays, with all the CRISPR-Cas9 stuff, maybe you can do it a little bit, but even now, it's really hard to do. So I said, look, my biology colleagues, they get it. Transcripts are interesting. There's some stories, but it's very hard to follow up on discoveries. Maybe we were slightly overselling the transcript thing, and maybe it's not quite really what you really want. And at the same time, another former student of mine, Nick Bray, who I acknowledged initially, got a math PhD with me, also decided to go into the wet lab. He's now a postdoc at the IGI at Berkeley doing also CRISPR-Cas9 stuff. And he said to me, in my group meetings, I'm like the computational guy, and everybody tells me, I want gene level DE. They don't want to give up the transcript level dynamics, but they want results at the end of the day at the gene level. And, I started thinking about this, and maybe I should listen to these former students of mine, even though they've abandoned math and gone into the lab. Maybe they have a point. And so Nick, started thinking about this and kind of started discussing with me, maybe we should really kind of think from scratch about what we're trying to do here and how one should reconcile transcript level versus gene level analysis. And so to do that, I want to just walk you briefly through a few steps of what really constitutes gene level analysis, which was really advocated for and pioneered with software programs like DSeq that are very widely used. This is a workflow from a paper that has 3 of the primary authors. Gordon Smith is the author of Limma. Maybe some of you have used that. Wolfgang Gruber is DSeq, and Mark Robinson did EdgeR. They wrote a protocols paper. And the point of this diagram is to show you that, it's a kind of a complicated workflow, but the real guts of it, or the relevant part is, you align the reads and you choose what program to use, and then you run this thing called feature counts or HT-seq. These are scripts that look at the alignments and just count how many reads map to a gene locus. And all these programs like DE-seq, EdgeR, they basically are all written actually in R to start with, because they take as input just these counts, just like a little matrix with, the different samples and their counts. And one of the things about cufflinks was we couldn't really do it in R because we had to do this whole really very presentation quantifying all the scripts. We had to work with individual reads. So the input to cufflinks was literally the raw alignments, which are huge, whereas here the input's just these numbers. So what does feature counts do? Well, as I said, it looks at a locus and counts the number of reads. There are actually different modes you can ask for. If you have a gene with multiple isoforms like this cartoon here, red and blue, you can ask to count just the things that are in there, a section of the two isoforms. But in any case, in a follow-up to the original cufflinks paper where we wrote more about differential expression in 2012, we tried to set up this cartoon where we tried to argue that it's really problematic to count in this naive way, even though it's convenient to feed things into R, in that, something like the average of the sum is not the sum of the average, something like that. And I won't spend too much time on this, but what we showed in this cartoon is, like, if you look at the first row there, condition A and condition B, there's the blue and red isoforms, and the little dots are the reads. In real life, they're not colored or painted for you, they're just reads. But if you knew really where they were from, as in this example, you'll see that there are 10 in each condition. So the actual log-fold change you'll get if you just sum them up is 0. But in reality, the abundance calculation, which is all the way on the right, gives you 0.32. And the reason for that is that the abundance of an isoform is not just the number of red reads or blue reads, but the number divided by the length, because the more sequence you have to sample from, just the more counts you're going to get. So what you really need to do is divide by length, and in one case you divide 10 by the full length, L. That's the right-hand condition B, because red has length L. Blue has length 2L, so for condition left, there are 4 reads that are blue and the rest are red, so you get 4 over 2L plus 6 over L. So what we showed in the thought experiment is that even though it's not just that the quantification is wrong when you just add up the reads because you're not normalizing by length correctly, it's also the case that wrong doesn't cancel out wrong. Because one of the arguments that were made about just taking the aggregate counts is that, maybe it's not quite right, but if you're just doing DE and you're just comparing two conditions, then wrong might just cancel out wrong. And so we tried to make this argument. The actual math, if you're interested, what it really boils down to is that you can't add up fractions by taking, a third plus 2/4 is 3/7. You're not supposed to add the numerators and the denominators separately. I call this the freshman's dream because I've taught calculus for many years, and the freshmen, they love this. They love to do things this way. It's kind of like in algebra, you do that. What I wanted to say is that sometimes it's actually right to do the wrong thing. Like, the thing is, if the denominators are equal, if at the top it said 1/3 plus 2/3, then it actually is okay to do this wrong math, because then wrong does cancel out wrong. And so, and that's actually often the case in math, like 1 plus 2 cubed is not 1 cubed plus 2 cubed, but sometimes it's true, right? If you're in the right field. And so the thing is that it's fine if you use DE-seq, because most of the time, or a lot of the time, isoforms have similar lengths, and so this issue is not always so bad. But it is wrong to do the wrong thing. And we actually looked at real data in our paper where we looked at a microarray experiment done on the same sample as RNA-seq. This was from the RIN lab, which Cole was working for at the time. And in this plot, the tails of the arrows are what you get with the naive counting, and the tips are what you get when you actually quantify at the gene level by first doing the isoforms and then adding them up. So, and you can see there's better concordance when you do that. So it actually matters on real data. It's not just a thought experiment. So, you know, we've advocated for this for a long time. And in our workflow, that's how we did things all along. And I was happy that, you know, finally, like a year and a half or 2 years ago, the gene people finally said, you know what, you're right. And they wrote a tool called TX-import, and that's what they advocate using now. TX-import reads in quantifications made by a program that actually estimates isoform abundances to add those up and produce gene counts from those abundances so that it doesn't have this issue. And that's the current workflow for a program like DE-seq. But the thing is that that still has some really fundamental issues. And I'd just like to walk you through some examples to show that. But first, I just want to show you exactly what TX-import does now when you run the standard workflows. And I thought I'd show it not conceptually, but on real data. This is actually data in the next couple of slides from a recent paper by Fromm, where they did an experiment in mouse looking at neural progenitor cells. And they actually did a couple of different, there's 2 sec, but the main thing is in the data we're comparing untreated mice versus cells that have been treated with dexamethasone. So it's sort of a simple condition A, condition B. And I just want to make illustrate the methodology by way of example. So what you see here is there's red and green or blue. Those are 2 isoforms, and then you have condition A and condition B. So what this program does is, on the first plot on the left, you're looking at counts. But first, it divides the counts that have been estimated for each isoform by the length. And what happened is the right-hand side you know, went up. I'm sorry, I think I said the wrong thing, and I'm on video, so I apologize. The red and the blue are the 2 conditions, and the left and the right are the 2 isoforms. What you see is the isoform, the counts have suddenly gone up when you turn them into abundances because it was a short isoform. So because it was short, it had fewer reads, but it's actually quite abundant. So that's the first step. And then once you're in actual abundance space, then it's legal to add up the abundances additively. And so that's what's happening here. And you get these 2 peaks now for the 2 conditions where you've added up red to red and blue to blue. And it's actually, that's apples to apples now because you've normalized the abundances by length. The problem now is that the right-hand side is in what are called TPM units, or transcript per million. That's some normalized version of counts divided by length. But programs like DE-seq, they do this thing called shrinkage or regularization. Because you have very few replicates, they kind of share information across genes to better estimate variance. And to do that, they actually need counts. So what happens next is that these abundances are converted back into counts, and that's what is now fed into the programs. And how do you go from abundances into counts? Well, you need a length for the gene. And this is kind of weird because the gene has 2 isoforms, and they have lengths. But how do you give a length to a gene? I mean, is it the average? Is it the max? Is it the min? So nobody knows, okay? So what they actually do is they depending on the mode that you use, you might take the median or the mean, they just do something, and they get some counts, okay? And that's how they share information. Genes that have similar counts this, produced this way, share information about variance. And so this is problematic. And there are actually a number of issues. So we tried to sort of classify them, and I'll try to go through them fairly quickly. There's what we call variance distortion, and then there's masking of transcript dynamics. So So let me talk about variance distortion. So when you use the mean of the lengths to multiply the abundances to go back to these, I don't want to call them pseudo counts, but there's some pseudo something, they're not really real counts. The heights of red and blue are actually pretty good. The effect sizes have been correctly measured, but and everything is scaled correctly, so the units don't matter so much. But the variance is problematic now, because when you have replicates and you're now doing this weird transformation for reasons that I won't work out the math for in a slide, but as a good homework exercise, the variance estimates are all off. And if you want to just think about it intuitively, you started out with actual counts. And in the simplest case, those counts, their variance should look something like their mean, assuming there's no biological variance. But now you're back in the count setting that's totally different in some other, scaled way, and you haven't really correctly scaled the variance. Okay? So at a high level, that's the problem. And so that's one thing that's not good. But then there's three other issues which have to do with the fact that you've summed up the isoform abundances, and I call them cancellation, dominance, and collapsing. And this is real data, so I'm not just making these up conceptually. This is from this very experiment. Here you see a case where one isoform went up, one isoform went down, so they sort of cancel. Okay? At the gene level, after you've gone through this whole thing, everything looks equal. So no way you can identify differential expression here if you only look at the gene counts. And that's what I showed you even back from the cufflinks example. Domination is a slightly more subtle issue where you have one very highly abundant isoform that dominates where there's real change. But now you go through this whole thing, and also because you've messed with the variances, you get a little bit of a difference between red and blue at the end, but it's not big enough to be significant. So basically, the very abundant isoform has dominated an effect of a less abundant isoform. And actually, all these examples were taken not just from real data, but from an analysis where they really were not, things were not identified as differential at the gene level. Finally, there's collapsing, which at its heart is got to do with this variance distortion, and I won't go into too much in detail, but here you have maybe three isoforms in this example where each of them moved a little bit, and looking at three changes that occurred in the same direction, you might conclude that this thing is actually differential, but the variance distortion has masked this and essentially collapsed this change. So these are the kind of scenarios that occur. This one is very common, actually, because it's quite frequent that transcriptional changes drive changes in every isoform, even though they might be small. It's not so simple to solve these things by switching to an isoform level analysis, because there, these effects might be very small and not detectable. In aggregate, maybe you could have seen them. And second, when you test for isoform DE, you're testing an enormous number of isoforms, and so you lose a lot of power, because when you do a multiple testing correction, you're got to get more significance on any individual event than if you only tested 20,000 genes. The human annotation in Ensemble has close to 200,000 isoforms now. So you're testing 10 times more hypotheses, which is a huge loss of power. Okay, so we thought about how to address this, and we came up with an idea that's actually very basic and simple. It just hadn't been used in the setting. So what's the idea? Well, instead of first quantifying the abundances and adding them up to get gene-level abundances and then doing DE, our idea was, let's do DE on individual isoforms, but not lose all this power by aggregating the p-values by gene. Kind of like the way you should think about this is, if you think about just one gene, each isoform test is like a study where you did a test and you got a p-value, And now you're asking in aggregate, looking at all these p-values, did I see something different in this whole gene? And this comes from a literature that goes back to the 20s. There's a thing called Fisher's method. You know, everything is called Fisher's method in statistics, and Fisher did a lot of great things. This is one of them. This is his least known method, actually. So this is the following thing. You've done like a number of studies, let's say for cancer, and you've got p-values for smokers versus non-smokers dying, let's say. And you want to take all your studies and aggregate those p-values to like decide, is there really a problem here or not? And so what Fisher observed is that if you've done 10 studies and you've got 10 p-values, then under the null hypothesis, this is really what the p-value means, those p-values should be uniformly distributed. So you're like looking at 10 numbers that should be uniform between 0 and 1. So he devised a test of that fact. Let's look at these 10 p-values and say, are they really uniformly distributed? And so his actual test is to multiply them and take the log, and that should be chi-square distributed. It's a method. Since then, there have been other methods, and although they're used for meta-analyses like I described, they're generally not used in bioinformatics. They have been used for GWAS, actually, where people associate SNPs with disease, and they want to actually aggregate the p-values in a gene, because they don't necessarily care exactly about the SNP and get more power that way. But in RNA-seq, not so much. So one of these methods, which you might try, I call CIDAC aggregation. And I'm going to mention this method first because there is one single program I'm aware of called DEX-seq. Maybe some of you use it. It looks for differential exon usage, and it used something like this method, although not quite right. And I'll talk about that in a second. So CIDAC aggregation is actually a very intuitive idea, which is, for this kind of aggregation, is you get p-values for all your transcripts. And we actually have a method to do that, by the way, which is, we published a paper earlier this year on a program called Sleuth that its sole objective is to be good at getting transcript-level p-values. What you're going to do is, with your, let's say, your 10 p-values for the 10 isoforms, you're going to ignore them, but just look at the smallest p-value. The idea here is, let's see if there was at least one transcript that was significant. So you take the minimum, and what you should do is adjust this minimum p-value with this formula 1 minus the minimum to the power of the number of isoforms, and this transform comes for the, it's sort of analogous to what's called the CIDAC correction. That's kind of like the Bonferroni correction. And the point here is that if your p-values are uniformly distributed, and you take the minimum 1, and you do this transformation, then that transformed minimum p-value ought to be uniformly distributed. And you can test for it. So that's a little lemma. You're going to get a new meta p-value for each gene, and then you just correct those across all genes. And now, what your gene-level analysis will do is it'll give you a gene back as an answer for being differential, but really what it's saying is that under the hood, at least one isoform actually was differential in it, but it doesn't tell you which one. Okay? So DEX-seq did this kind of thing, but they actually did something sort of interesting, and I don't know too many examples. They didn't rank this according to these transformed minima, they just did it according to the raw minima. And they have a way to control the false discovery rate. And it took us a while to like untangle this math, which I won't do in front of you today. But it turns out that this is correct, actually, what they did, in that their method controls the false discovery rate, but it's wrong in that it's not what you want to do is rank things by the minimum. You want to rank them by the transform minimum. So it's a kind of case where it's quite subtle, where the wrong thing is correct, but also wrong, something like that. I mention it just because in the slides that follow, I'm going to compare it to this method, but I'm going to compare it to the correct method instead of this slightly weird thing. Okay, so we looked at some simulations. I tried this minimum method out. It actually did slightly worse than standard gene level DE, where you first sum the quantifications and then do DE. And I'm showing you the results where we plug things into Sleuth, which is our program, but we have a bioarchive preprint now where we also plug things into DESeq2, and you get similar things. So why is that? Well, one of the problems with the MIN approach is that it's good in the isoform switching case, cancellation, but it doesn't do well in the setting of collapsing. You know, that was where everything moved a little bit in the same direction. And you can see why that is, because you might have two p-values that are not significant. Maybe they're 0.09 and 0.09. And if you use this minimum, well, it still doesn't look significant. But together, 0.09 and 0.09 are actually significant. And actually, in fact, with Fisher's method, aggregating 0.09 with 0.09 gives slightly under 0.05. So, so this is showing this on an actual real data set where you can see, I've circled in the bottom here, none of the actual p-values are significant for an isoform, but Lancaster's method, I'll talk about in a second, it's a modification of Fisher's method, can identify this gene as actually differential. So what is this Lancaster's method? Well, so Fisher's method, the actual statistic you compute is you sum the log of the p-values. If you'd like, it's actually a good exercise for fun. Tomorrow's Friday, so Friday nights are great for this kind of thing. You can take two p-values, p and q, and Fisher's method produces the merged or aggregated p-values pq 1 minus log pq. But the thing is that in this meta-analysis conception, p and q are equal. You know, you did two studies and they both produce p-values and they're equal to each other. Lancaster posited that maybe one study had a lot of data and maybe one study didn't. And so maybe you should not treat the p-values equally and you should weight them instead. In the RNA-seq case, that's actually relevant because some isoforms have a lot of counts and you can be fairly confident about them and some don't. So, so Lancaster's method is a weighted method. If you're curious, it basically works by, instead of taking the log, which can be viewed as transforming the p-value into a chi-squared value, sort of the inverse transform, you do the same thing, but you transform it not to a chi-squared distribution of degree 1, but of a higher degree, and that's kind of how you weight things. So for today's talk, you can just think about it's like a weighted Fisher's method. So this is actually a simple experiment. If you just take two p-values that are the same, just to prove it to you, then this minimum value thing, the SIDAC, is not as good as Fisher's. Because of the exact reasons I've described. Okay, so let's look at some simulated data. So I should say this simulation is from the Sleuth paper. It was designed by taking a real data set, actually Javadas data, where we took two populations and we learned effect size and the kind of changes that happen in real data and then simulated that into a data set at the isoform level. And so what you see here is that the SIDAC correction is slightly worse than the gene level, as I've shown you before. Red is just the vanilla gene DE, and green is slightly better. But this kind of plot, so, you know, the x-axis here is the false discovery rate. Like you've just ranked every method is given a ranking of genes. You're going from the top to the bottom, and as you scroll down the ranking, you're asking, how many false discoveries do I have, and how many true things did I find? That's the y-axis. And it doesn't look like a big change, but it's actually huge. So at the kind of false discovery rates you care about, like 0.05 or 0.01, we're finding like 2 or 3 times more real genes. And I was really like shocked when we got this result because I've actually seen many times talks and on forums, people saying that, you know, gene level DE is basically solved. I mean, there's like 50 different programs. They all shrink differently or do this a little bit differently or that a little bit differently. But basically, people have had the sense that, yes, the programs give different results, but they can control the false discovery rate and they have some sensitivity, and that's the best you can do. Well, I actually think you can do a lot better. And so when we got this result, my student Lynn So the thought, look, we got a huge improvement by basically agreeing to stick with the gene level, but doing it by taking apart the gene into pieces and doing DE there. And then she said, why don't we take that a step further? Because actually, to get transcript level quantifications, we go through the step of actually doing this EM algorithm and all sorts of fancy statistics. Why don't we aggregate prior to that? And I'd like to explain that concept to you. In particular, at least in my group, the way we get quantifications these days is we use our program Callisto. And one of the things about it is that it has this concept of prior to quantifying, generating what we call TCCs. Or let me explain what that is to you. So in the plot on the top here, I have a gene with 3 isoforms, and I have a couple of toy reads on top. And what Callisto does, it does this thing called pseudo-alignment. It doesn't try to exactly align the reads. I mean, here they're aligned, but Callisto doesn't do that. It just tries to produce the output that's at the bottom right. What it does is it tries to just measure which transcripts are the reads compatible with. So let's go through this. In the very first column, green up at the top, there's a little read, and it's actually ambiguous between transcript 1 and transcript 3. And so that gives us output at the end that there's a set, T1, T3. And we saw it occurring one time. So the pseudo part about the alignment is that we're not telling you where reads align, just what they were compatible with. And if you move one over, you see those 2 reads were compatible with T1 and T2. And that's the next row on the bottom right. T1, T2, T3 are the reads that were ambiguous to all the transcripts. And you'll see that there were 5 of those. And so we call those sets equivalence classes, because that's what they are. Reads are equivalent to each other within those equivalence classes. But we call the counts sort of transcript compatibility counts, or TCCs. And when you run Callisto, it actually can output this data. And then we take this information and we go ahead and quantify the transcripts. And kind of one of the insights of the Sailfish paper that preceded Callisto was that basically this is the sort of information that's relevant for the models that actually quantify RNA-seq. So that's how Callisto works. So Lynn's insight was, let's leave the quantification out of it because we introduced a bunch of noise there. Let's just get p-values for these sets. So we got condition 1, condition 2. We have some counts for T1, T3. We have some counts here for T1, T3. Are they different? That is a really interesting idea because T1, T3 as a set is not particularly biologically meaningful, but we don't care about biology right now. We care about statistics. Well, the biology comes out of a p-value for the gene. And we all love genes. That's good. So it's an interesting idea. So we actually implemented this, and I'm really pleased with this figure. So on the left-hand side, I have on this real data set the top genes produced by this aggregation of the p-values that come out of TCCs. And on the right-hand side, I have the p-values that come out of aggregating the transcript p-values. And I put little red lines so you can see that it's the same genes. I mean, they're like a little bit different in order. Just to convince you that, I mean, this is kind of shocking because I believe it's the first ever differential expression analysis that was done without doing anything. So, think about this, right? Like you got reads, and we didn't align them. Or we didn't actually do an alignment. We pseudo-aligned them. All we learned about them is which transcripts they're compatible with. And we can do that really fast with Callisto. And like this part of Callisto takes a minute or maybe 2 minutes on 20 million reads on the laptop because you're doing nothing at all. And you take that, and then you do nothing, and you get the gene list at the end. That's pretty cool, right? Look, Ma, no hands. That's the idea. All right. Just to give you like another cool example, you can do your goal-level enrichment analysis this way. I think this is the first ever goal-level enrichment analysis that was done without alignment or without quantification. What I'm showing you here is this data set. This was dexamethasone treatment of these neural progenitor cells, and that should affect immune genes. And so we looked at the p-value for enrichment in the standard Go analysis with the standard gene level analysis, where you get your genes out of, just the standard way, or where you aggregate by Lancaster, Sidak, or these TCCs. And you can see that the TCCs are actually really good. Where are we really able to do the analysis without quantification? Also on simulation, we find that the purple, the TCC part, is very comparable to using transcripts. And actually, the reason we're probably slightly worse here is sometimes you have TCCs or equivalence classes that have reads ambiguous across genes, and it's not what you do with the p-value. So for this analysis, we tossed that data out. We actually tossed out like 12% of the data here. And we know we have some ideas of how not to do that, but we just felt it was amazing that we could even do that and still get very good results. And I promised you earlier that I presented sleuth, not just because it's our program, but because when we did these analyses where you plug, you could produce all these p-values with DSeq2, whether you're doing TCCs or transcripts, you get similar orderings according to method, just slightly worse results. But so it's not specific to sleuth here. So you might ask, why would you do your DE analysis with these weird TCCs? Well, one reason is it's not just cool. It can actually give you information that you can't get right now. And just to give you an example, which involves, it's a 3' assay where your RNA-seq is only reads from the 3' end of a gene. And there are many such assays with 5' end, 3' end. First of all, the TCC analysis of such data, well, let me go back for a second and say, when your protocol looks like this, you cannot run a program like Callisto or Cufflinks, because those programs assume that the reads are uniform across the transcript. But you can use the TCCs, and just to show you that, because I'm a little bit nervous that biologists will say, look, there's no way we're trusting this stuff. Like, what are TCCs? What are you talking about? I just want to show you that, the p-values you get out are very similar to what you get if you just sum the counts up. So we're not doing something totally crazy here. But we can actually extract cases of alternative 3' usage. Not always, but sometimes we find examples where there's an alternative 3' end that led to a unique equivalence class. And we can now see that, that difference in that equivalence class comes out, calling the gene differential. This is completely masked if you just add up the counts of the gene. So this is one example from a data set from Dolan et al., which was just published earlier this year. Going back to the Go analysis, we realized that we could actually do something interesting. Instead of this kind of enrichment analysis where, you get genes coming out of your DE and then you put them in Go software and it says, okay, they're enriched in this term. So why aggregate at the gene level? If you have a Go term you're interested in, and all the constituent transcripts and all their p-values and get a p-value for the whole Go category. There's no reason you have to aggregate to genes. You could aggregate to any level or for any kind of resolution that you want. And so we did that, and we call this a sort of perturbation analysis because it's a kind of new complementary type of analysis to what you might do with standard Go. So, and that's kind of demonstrated here. So each dot here, its x-axis is a Go term's p-value when you do a standard enrichment analysis. And the y-axis is the p-value that comes out of aggregation. And, intuitively, the way that they're different is, as you go up the Go hierarchy, it's more and more likely that some gene that belongs to your high term was perturbed. So you're more and more likely, actually, to get low p-values as you go up, which is the opposite of enrichment analysis. But it provides you, interesting information. It's telling you that, if you're interested in a pathway or a specific class of genes, it's just a measure of, was something perturbed in this category? And one interesting thing that came out is when you look at the categories that came out significant in the perturbation analysis, they are significantly enriched for the word immune, and that just does not happen to be true when you do a standard Go enrichment analysis with this data. So I think this will be an interesting kind of resolution at which you would like to do aggregation. Okay, so I think I'm pretty much out of time and I'm out of slides. I'd just like to make some final comments at the end. So one thing is that gene level analysis doesn't mean you have to abandon transcript level results. There's a really interesting paper that just came out very recently, actually, where it's discussing a two-step method where you kind of save some of your false discovery rate budget when you're doing your gene level analysis so that you can then go back into those genes and look at for individual transcripts there. And the kind of method I just proposed, which really is ultimately very simple, is compatible with that. So you can do your gene level analysis the way I discussed. Actually, in this paper, they used the DEC-seq min thing to do that step. So that's one thing I wanted to say. I also wanted to say that aggregation can be performed with numerous groupings of interest, not just levels like genes to go, but you can group, for example, transcripts by the transcript start site, like we did in the cufflinks case. And I think that will be interesting for kind of measuring perturbation inside groups that have biological meaning. One of the things that we plan on doing soon, we have a website called the Lair, where we have a kind of snake-based workflow for automatically downloading and processing data from papers and doing automatic differential analysis. So it will be interesting to go back. We have 15 papers loaded there right now to redo all these analysis with that method, and you'll be able to try out this approach through Sleuth in a release that we'll put out soon that will do these things. Okay, so I think I'm out of time, and I'll end. I'll just like to thank Lynn and Nick and Harold again, and thank you all for listening. Okay, that's a really interesting question. So, so we looked at that very carefully in a second, because it turns out that I said was somehow wrong. It has that effect, that it will preferentially call out, which way does it go? It will preferentially select for genes that have many transcripts. And the reason is obvious, because they're using the raw min value, and the more transcripts you have, more isoforms, the more likely by chance that you'll get a very small p-value. These other methods don't, I think, have that sort of bias, because at the end of the day, under the null hypothesis, they're simply measuring that the p-values come out of all the isoforms are uniform. So, so that's, yeah, so.
Can you talk about transcript disannotation?
Yeah, so I'm asked about this a lot, and you know, in the setting I just discussed, as in the Callisto framework, your input is an annotation of transcripts which are known, and that's sort of okay in human. It's not so okay in other organisms, and we have done analyses, for example, measuring what happens when we drop an isoform. And, you know, it's complicated to answer what will happen. I mean, it really depends on the gene. One of the things that we were talking about this morning with Cedric and some others is that when you do pseudo-alignment, we often can see in the pseudo-alignment hints that we are actually missing annotation. And we've had in mind for a while to, like, flag those transcripts or genes and, you know, couple this with assembly methods to go and try to recover those isoforms. And I think that's a really good project to do. We haven't done it yet. So the results have that caveat right now. But it's a very good question.
How does it handle ambiguously map read between separate genes instead of just separate
Right, so in the results I showed now, when you're getting a transcript p-value and aggregating transcript p-values, it's a non-issue because Callisto has statistically assigned those reads. And so it's given a fraction to this transcript and a fraction to that one in the other gene. And so somehow that all comes out in the methodology. However, if you're trying to just use the TCCs, that's a real problem because the TCC itself is ambiguous between two genes. And because you haven't quantified, you don't necessarily know how to split apart the counts that go with that TCC. So we don't have a really great solution to it yet. We've tried apportioning the reads uniformly, and that hasn't worked so well. But we have some ideas of how we can do a very crude-ish quantification to kind of split the TCC in some meaningful way and not throw out that data. Another idea, which I think makes sense, is that in such cases, you may wish to actually report the p-value on the aggregated gene set. Because, you know, often in those cases, you're looking at a gene family with many, duplicates of the gene. Maybe it's meaningful to just report a p-value for the whole set, and that's really compatible with this method, and I like that idea, actually. But that involves a bit of sort of UI for the user. So that was a good question as well. Yeah.
That's sort of a related question, which is, what do you think of quantification at exon level instead of gene level?
Yeah, so I mean, DEX-seq actually is doing exactly that. It's counting reads on exons. And the min of that is done there was actually across the p-values that come from exons. In this new paper I mentioned with the two-step procedure, they actually used DEX-seq differently, but the original DEX-seq was doing that. The problem with that is that you're throwing out a lot of data because all the reads that cross junctions are being discarded. But I think there are instances that will be biologically meaningful to do that, and this is totally compatible. Basically DEX-seq was doing exactly that. But I don't like to do the analysis usually that way just because you're throwing out all the information across the whole gene, of all the junctions, which is a lot of, reads are, like basically, the longer the reads get, the worse it is to do that kind of thing, right? If you think about it, right? Because your reads start crossing even multiple junctions, and now, what do you mean by an exon count? Is it all the reads that cross that exon, but now the same read is giving you data about two? I don't know, it gets complicated. Thank you.